Cellular respiration is a set of metabolic reactions that convert energy from nutrients into ATP and releases waste products. The equation for cellular respiration is C6H12O6 plus 6O2 yields 6H2O plus 6CO2. There are 4 steps. Glycolysis happens in the cytoplasm of the cell. The Krebs cycle or citric acid cycle, pyruvate oxidation, and the electron transport chain occur inside the mitochondria.
As an alternative to cellular respiration of organic substances, fermentation is a method of harvesting ATP without the need for oxygen or the use of the electron transport chain. There are many types of fermentation, but we will focus on the 2 most common types, alcohol fermentation and lactic acid fermentation. Alcohol fermentation takes place in the cytoplasm of many bacteria under anaerobic conditions. In alcohol fermentation, pyruvate resulting from glycolysis is converted to ethanol. Firstly, during glycolysis, 2 ATP and 2 NADH are produced as well as 2 pyruvates. CO2 is released from pyruvate, which converts to acetaldehyde. From there, acetaldehyde is reduced by NADH, gaining electrons to form ethanol. This step regenerates the supply of NAD+ needed to continue glycolysis. Lactic acid fermentation takes place mainly in the cytoplasm of human muscle cells. In lactic acid fermentation, pyruvate is reduced directly by NADH to form lactate. In glycolysis, 2 ADP and 2 phosphate groups are converted to 2 ATP via substrate-level phosphorylation, and 2 NAD+ gains electrons from the glycolysis process to form 2 NADH and 2 protons. These electrons in NADH are then transferred into the 2 pyruvates, reducing the organic compounds and forming lactate. Oxidizing NADH regenerates NAD+, creating a cycle of NADH and NAD+ to power glycolysis.
Glycolysis occurs in the cytosol of the cell's cytoplasm. To begin, 2 ATP molecules are used to break bonds in a 6-carbon sugar-glucose chain, releasing 2 ADP molecules. The glucose molecule then splits into 2 3-carbon sugars, which are then oxidized. 2 NAD+ are reduced to 2 NADH by electrons released from the oxidation of glucose. 4 ADP and 4 phosphate transform the 3-carbon molecules into 2 pyruvate and release 4 ATP and 2 H2O. This process creates a net of 2 pyruvate plus 2 H2O, 2 ATP, and 2 NADH plus 2H+.
Pyruvate oxidation. The mitochondria is composed of 2 membranes, the inner and outer mitochondrial membranes. The inner membrane has infoldings called cristae, which separates the interior into the intermembrane space and the mitochondrial matrix. Inside the mitochondrial matrix, pyruvate undergoes a series of reactions to remove carbon dioxide and form NAD from NAD+. The resulting compound from these reactions is acetyl-CoA, which then enters the Krebs cycle for further oxidation. Krebs cycle. The Krebs cycle occurs in aerobic organisms within the mitochondria. Acetyl-CoA enters the cycle and combines with oxaloacetate, a 4-carbon molecule which yields citrate, a 6-carbon molecule. Essentially, citrate is then decomposed to regenerate oxaloacetate, with the release of CO2 in the process. As a result, 1 ATP is formed, 3 NAD+ are added to 3 H+, to create 3 NADH. And 1 FAD binds with 1 H+ to produce 1 FADH2. ATP is generated from substrate-level phosphorylation, which occurs when an enzyme transfers a phosphate group from a substrate molecule to ADP. 2 cycles have to occur since each glucose produces 2 pyruvates in glycolysis. After 2 cycles, the products are 4 carbon dioxide molecules, 2 ATP, 6 NADH, and 2 FADH2.
The electron transport chain consists of molecules embedded in the inner membrane of the mitochondria. The folding of the inner membrane, or cristae, increases its surface area. Therefore, many electron transport chains exist in each mitochondria. Embedded in the inner mitochondrial membrane, there are 4 complexes labeled with Roman numerals 1 through 4. Electrons from NADH enter complex 1 and go through until it reaches complex 4. FADH2 adds its electrons from complex 2 at a lower energy level than NADH. During the movement through the complexes, the exergonic flow of these electrons is used to pump hydrogen ions into the intermembrane space, creating a concentration gradient of H+ across the membrane. Electrons from NADH and FADH2 move from a lower affinity to a higher affinity, and a hydrogen ion is removed and what's left is NAD+ and FAD. H+ flows from high to low protein gradient through ATP synthase and powers ATP production. When energy is stored in the hydrogen ion gradient and is used to drive cellular work in the ATP synthase, this is called chemiosmosis. Protons bind to the catalytic site, and once activated, ATP is produced from ADP plus phosphate. The electron transport chain also produces H2O when two hydrogen ions bond to the oxygen atom after being released from the ATP synthase. The oxygen atom is referred to as the last final electron acceptor because it combines the electrons leaving the electron transport chain with H+ to form H2O. This method of producing ATP is named oxidative phosphorylation because it is powered by the redox reactions of the ETC. The electron transport chain produces water, 32 ATP, NAD+, and FAD.